Hello everyone, I'm Suganya, Assistant Professor from PG and Research Department of Biotechnology in Arinarana College. The topic I'm going to be talk is about cell cycle and cell division. Cell cycle. Cell cycle is the basic structural and functional unit of any living things. It also known as fundamental building blocks. A cell comprises several organelles such as cytoplasm, cytoskeleton, endoplasmic reticulum, lysosomes, nucleus, ribosomes, etc. The multicellular organisms grow by the addition of new cells, and the cells can be arise the division of free existing cells. In this, cell divisions is fundamentally similar to the all organisms. It is defined as unity of life. Phases of cell cycle. A phases of cell cycle, it indicates somatic cell exists in two main state. The first stage is interphase, and then second stage is mitotic phase. The interphase is a long undividing state, and then mitotic phase is a short dividing state. It also indicates M phase. Interphase. This interphase cells grows by synthesizing biological molecules such as carbohydrates, lipids, proteins, and nucleic acids. This interphase take place 10 to 30 hours. This divided into three types. First one is first gap space, second one is synthetic space, third one is second gap space. The first gap phase indicates G1, synthetic space indicates S, second gap space indicates G2. This is the cell cycle. In the cell cycle divided into two categories. So first type is the interphase, second type is the mitotic phase. In this interphase, it indicates three units are there, G1, S, G2. Then mitotic cell division is this and mitosis, cell division, nuclear division, and then cell division occurring by the cytokinesis. G1 space. In this G1 space, gap 1 space, and then G1 is the initiation of replication in genetic material, and this during this phase metabolically active. The gap between previous mitosis and beginning of the DNA synthesis. And this G1 space takes carbohydrates, lipids, proteins, and RNA are synthesized. No change occurred in the DNA content of the cell. And then muscles and nerve cells never divided again and remain in G0 space. In this G0 space is also known as cell resting space. S phase. S phase is also known as DNA replication takes place. These cells is denoted as 2n number of cell division. It is indicates after replication becomes 4n number of cell divisions. In this S phase takes place 6 to 8 hours. G2 space. In this G2 space, a gap between DNA synthesis and nuclear division, and importantly RNA transcription and protein synthesis can be continuously synthesized. The centrioles and mitochondria and Golgi apparatus are doublet. In this G2 space, during this phase, DNA replication and RNA and proteins and macromolecules are required for multiplication of cell organelles. This cell prepares to go into mitotic phase. In this G2 space can take place 2 to 5 hours. Mitotic space. Mitotic phase is an equal division of cell undergoes a completion reorganization techniques, and this mitotic phase is orderly distributed of the duplicated of the chromosomes into the daughter cells. The nuclear envelope breaks down and chromatin is packed into the visible chromosomes. And the endoplasmic reticulum and Golgi apparatus break down into small vesicles. This can be cells enter into developed into process of the cell divisions. Mitosis. So mitosis is a common method of cell division in eukaryotes. It occurs in somatic cells. It takes place to the 30 minutes to 3 hours. Mitosis. In this mitosis can divided into two groups. So first one is the karyokinesis and second one is the cytokinesis. Karyokinesis is take place to four types are there. So first one is the prophase, metaphase, anaphase and then telophase. And this cytokinesis is an cytoplasmic cell division take place. So karyokinesis is an method cell divisions. The nucleus splits into two daughter nuclei. Prophase or early prophase. The cells becomes less rounded and cytoplasm turns more viscous. This is microtubules can be assembled around the centrioles. And these two pairs of the centrioles to start moving opposite ends of the cells. Metaphase. In this metaphase take during time, so 2 to 10 minutes, the chromosomes move to the equal plane of the spindles. And this chromosome soon get aligned to the middle of the spindle in plate called metaphase plate. And these two chromosomes fibers can connect to the sister kinetochores to the opposite poles. Anaphase. This phase chromatids each of chromosomes slightly separated to primary constructions. Their kinetochores stretch towards the opposite poles of the spindles. This anaphase takes to the VJI shapes. So depending upon the position of the kinetochores. In this anaphase ends of the chromatids each of the opposite poles. Telophase. In this telophase, long and complex phase and chromosomes to pole unfold of the long slenders. It is a nuclear envelope reconstructed around the each chromosomes. The envelopes is the surrounded to entire the set of chromosomes. It can be organized to the cytoplasm matrix of the nucleolus. This is the telophase. In this telophase, to begin to disappear the depolymerization of the microtubules. And this only short microtubules are left. The centriole takes up the characteristics of the interphase positions. Karyokinesis. This karyokinesis is the type of meiosis cell cycle, and these two daughter nuclei formed to telophase are identical. This karyokinesis depends upon the nuclear splits into the two daughter nuclei. The arrangement of spindle microtubules can be poles of the cells. The connection of the chromatids to each chromosomes, the opposite poles of the spindle to ensure the opposite poles. It is an primarily stage of cell division in mitotic stage. Cytokinesis. Cytokinesis is the in this phase cytoplasm of the cell division with mitosis ends. The cytokinesis occurs in plant and animal cells differently, and this results into two genetically identical of daughter cells. These cells typically divided into process of cleavage. And the short spindle microtubule becomes a structureless material called midbody. And this appearance of the plasma membrane is level to midbody. It is and gradually deepens. This membrane fuses with the cytoplasm matrix to two daughter nuclei. It is an independent of the daughter cells. It is an cleavage and then contractile ring. This indicates that independent of the daughter nucleus into two daughter nuclei. This enter to the G1 space on next cell cycle. Meiosis. Meiosis is an particular cell takes place a particular time. And the cells are sexually reproducing organisms undergo meiosis. And this gametes in animals and spores and higher plants. And this meiosis take place meiocytes. It indicates oocytes, spermatocytes, sporocytes. Meiosis is also known as meiosis 1 and meiosis 2. So meiosis 1 is takes 4 phases are there. So first one is in prophase 1, metaphase 1, anaphase 1 and telophase 1. Prophase 1. This is more complex than mitotic prophase because of recombination occurs. It can be 5 sub stages are there. First one is on lipotine, pachotine, zygotine, diplotine, diagenesis. Metaphase 1. This can be cytoplasm move to the equal spindles. Two parallel metaphase plate. And each homologous chromosome has two kinetochores, so both are connected to the spindle pole. The two kinetochores of homologous chromosomes can be functional unit in metaphase 1. Anaphase 1. In this anaphase 1, homologous chromosomes can be separated, while sister chromatids remain associated at their centromeres. Telophase 1. The nuclear membrane and nucleolus reappear at the cytokinesis to the diet of the cells. In many cases of the chromosomes do undergo so not reach the extremely extended state of the interspace of nucleus. And the stage between the two mitotic divisions can call interkinesis. It is generally a short life. And interkinesis is followed by prophase 2. It is a much similar prophase than prophase 1. This is the telophase 1. It indicates nucleus reappear cytokinesis. And this take place the nucleus membrane to reforms around two haploid nuclei. Meiosis 2. In this meiosis 2, prophase 2, first stage is in prophase 2. Meiosis 2 is initiated immediately after the cytokinesis and before the chromosomes have fully elongated. This meiosis 1 and meiosis 2 is resembles of a normal mitosis, and then in this membrane disappear by the prophase 2. The chromosomes again become compact. Metaphase 2. In this stage, chromosomes align equal of the microtubules of opposite poles. The spindles can be attached into the kinetochores of the sister chromatids. Anaphase 2. Anaphase 2, it is stimulatively a splitting of the centromere of each chromosomes, which holding of sister chromatids together. And in telophase 2, this meiosis ends of the telophase 2. Two groups of chromosomes can one again get enclosed to the nuclear envelope. So cytokinesis follows, resulting in formation of tetrad that known as 4 in number of cell divisions can be divided into 4 haploid daughter cells. This take place meiosis 2, and this division meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2. In this telophase 2, cell divisions can be 4 in number of cell divisions can be update, and this haploid is number of n of unduplicated of the chromosomes. Significance of meiosis. Meiosis is mechanism which conversation of the specific chromosomes and then across generation of sexually reproducing organisms. And this even process paradoxically results in reduction of the chromosome number of half of. It also increases the genetic variability in the population of organisms from one generation to another generation. It is the variation of very important for the process of evolution techniques. It is an important to the significance of the cell division and cell cycle. Thank you.